So hello everyone, welcome to the video. Today here we will be discussing about the glycolysis, its pathway, and we will try to understand the simple step by easy explanation. And please watch the complete video to get a proper output of the video and how the basic concept of glycolysis works. So before starting, let us understand why glycolysis is important. Okay. So glycolysis is it is a pathway. It is a pathway that ensures the conversion of glucose into pyruvic acid. But why is it important? It is important because we know glucose. It can be termed as a fuel to our body. It is the only fuel. It is the only fuel in the red blood cell that can be used. Okay. What will happen if glycolysis will not occur? What will happen? Any idea? Basically, mature red blood cells are not capable of aerobic respiration. Okay, the process in which the organism convert energy in the presence of oxygen, and glycolysis is the sole source of the ATP here. So if glycolysis is basically not done, or it is interrupted, or it is not completed in a way, then the cells, they will lose their ability to maintain the sodium phosphate pump, and eventually the cells will die. And if the cells will die, you all know, the functioning of the body will be disturbed. So glycolysis is the basic metabolic pathway which is very important in the human body, or we can say in mammals. Okay, so let us start. So introduction, first of all we will understand what is glycolysis. In glycolysis, as we know that the glucose is a six membered ring of molecule. It is found in the blood, in the red blood cell basically, and usually it is result of breakdown of carbohydrates into sugars. Basically the carbohydrates are breakdown into sugars. So here we will see, look, this is the mitochondria. Inside here, this is the mitochondria, this is the cytosol. Okay, so this is the cell, total cell, this is the zoomed out portion. So the glycolysis occurs in the cytosol. This is glycolysis, it occurs in the cytosol, and two different cycles, citric acid cycle and oxidative phosphorylation, they occurs inside the mitochondrial membrane. So why it occurs in the cytosol? Basically because the glycolytic enzymes are only found in the cytosol, so it helps the process to complete easily. So we know glycolysis, it is a metabolic process. It may be termed as the foundation of aerobic and anaerobic cellular respiration, and we have seen it occurs in the cytoplasm. Okay, these are the points written here, nothing more. These all points are written here. Okay, and it enters the cell through a specific transporter protein. How it enters the cell? Here it enters the cell through specific transporter proteins. What they do, they move it from outside the cell into the cell cytosol. This is their function, transporter proteins only. Okay, we all the glycolytic enzymes we have discussed, they are present in the cytosol. Okay, in the next video we will also discuss about the citric acid cycle, its functioning, its importance, and the oxidative phosphorylation also. These all are interrelated to each other. So what happens in glycolysis? The six molecule of glucose. Here the six molecule of glucose are oxidized to generate three carbon molecule of pyruvate. So pyruvate, we all know pyruvate is formed, but the six carbon molecule of glucose, it is converted to three carbon molecule of pyruvate. So now we will see stepwise. So there are basically 10 basic steps in glycolysis. The 10 steps completes the full process. So we will see all the process one by one. Okay, first one, here the glucose, we all know is 6 carbon molecule. Okay, the 6 carbon molecule glucose is converted to glucose 6 phosphate with the help of hexokinase. This is the enzyme hexokinase. It is the enzyme that catalyzes the glucose into glucose 6 phosphate. So what happens here? The phosphorylation occurs here. Okay, this is the phosphorylation occurs. So what happens? The phosphate group is added, and it is derived from the ATP. So one ATP is also consumed in this process. In the process of phosphorylation, one ATP is consumed during the process. Okay, minus one ATP. So here magnesium ion. The magnesium, it is the atomic magnesium. It helps to shield the negative charge from the phosphate on the ATP. So the ATP we will see here is consumed and ADP is formed here. Okay. So this is the first step. The glucose is converted to glucose 6 phosphate and 1 ATP is consumed in the process we have explained. So this is the second step after the first step. The glucose 6 phosphate is converted to fructose 6 phosphate with the help of phosphoglucose isomerase. So what does the glucose isomerase do? It is nothing more the rearrangement of the carbon and oxygen bond occurs here. Okay, nothing more than that. It is a reshuffling or rearrangement of the mm, 6-member ring to the 5-member ring. Here we can see it is a 6-member ring and here it is a 5-member ring. This is basically rearrangement of the ring, nothing more. Okay, so this is the second step. Now we come to the third step. What happens in the third step? The fructose 6 phosphate. In the third step, the fructose 6 phosphate, which we have obtained with the um rearrangement, the fructose 6 phosphate is converted to fructose 1,6 bisphosphate. Okay, it is converted with the help of phosphofructokinase, which we can call PF. What happens here? Here also an ATP is consumed and the phosphorylation occurs here as well. Okay, and the magnesium ion, it acts as a cofactor. The magnesium ion acts as a cofactor like it was acting in the first step. Okay, so the ATP is now one ATP is basically consumed in this step also, during the process of conversion from fructose 6 phosphate to fructose 1,6 bisphosphate. Here also minus 1 ATP is consumed in the process, third step. So till now 2 ATPs are consumed. Now comes the fourth step. Fourth step, the fructose 1,6 bisphosphate with the help of aldolase, it splits into two sugars. Two sugars we can see here, dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Okay, DHAP and GAP. It is a short form. So the aldolase splits it into two sugars, and DHAP and DAP. So this fructose 1,6 bisphosphate 6 carbon ring is converted into two different carbon rings. Okay, two different carbon chains we can say. So now we will go to the next step of the fourth one. uh, the fifth step here, the two uh, two chains that we have obtained here with the help of triose phosphate isomerase. With the help of triose phosphate isomerase, the interconversion occurs. DHAP and GAP. DHAP and GAP interconverts with each other. Okay, here the interconversion occurs of both the chains. Now what happens? The glyceraldehyde 3 phosphate is carried on to the next step. Okay, here in the sixth step, now this was up to the fifth step, now this is the sixth step. We can see up to the fourth step there were six carbon chain. Now we can see there are 3 carbon chains. Now in the sixth step, what happens is the glyceraldehyde 3 phosphate dehydrogenase. This is the enzyme glyceraldehyde 3 phosphate dehydrogenase. It works here. It dehydrogenates and add the inorganic phosphate here. Okay, it adds the inorganic phosphate here. So it converts into 1,3-bisphosphoglycerate. So in this step, two main things occurs. we can conclude here two main things occur in this step. Okay, first of all, the glyceraldehyde 3 phosphate, it is oxidized by the coenzyme nicotinamide adenine dinucleotide, which we call NAD. Okay, the NAD. The molecule is phosphorylated by the addition of free phosphate group. It is phosphorylated with the help of by the addition of free phosphate group in this step. So this was the first incident that occurred. The second one, what happens is the enzyme which we call this glyceraldehyde 3 phosphate dehydrogenase, it contains very improper structure. So the phosphate group attacks the GAP. GAP is glyceraldehyde 3 phosphate. It attacks the GAP and releases it from the enzyme to yield 1,3-bis phosphoglycerate. Okay, to obtain this. So it generates NADH and a hydrogen atom. So in this process, NADH and a hydrogen atom is generated. So this is the sixth step of glycolysis cycle. Now coming on to the next one, this is the seventh one. It is very easy to understand stepwise. The seventh one, what occurs here is 1,3-bis phosphoglycerate. The 1,3-bis phosphoglycerate which we have obtained, the phosphoglycerate kinase. The phosphoglycerate kinase, it converts it into 3-phosphoglycerate. 1,3-bis phosphoglycerate is converted into 3-phosphoglycerate in this process. So a loss of phosphate group occurs here. Okay, the phosphate group, it transfers the molecule of ADP. So what it does is it transfers the molecule of ADP and yielding the first molecule of ATP. So ATP here is obtained. What happens? ADP, the phosphate group will transfer to a molecule of ADP and a first molecule of ATP is produced in this step. Okay, since we have two molecules of the same. Okay, here we have two molecules, so the 1,3-bis phosphoglycerate, two molecules we have here, so the net outcome here is 2 ATP. So we obtain 2 ATP in this particular step. In seventh step, 2 ATP is obtained. Now let us come to the eighth step. The eighth step, the 3-phosphoglycerate is converted to 2-phosphoglycerate with the help of phosphoglycerate mutase. Here nothing much happens. A phosphate group, it relocates. This is the only step. This is the only main main thing that occurs in the eighth step. Okay, now after this we will come to the ninth step. This is the ninth step. The 2-phosphoglycerate, it converts to phosphoenol pyruvate with the help of enolase enzyme. It converts into phosphoenol pyruvate. What happens here? H2, water molecule is removed. It dehydrates the 2-phosphoglycerate. So here 2-phosphoglycerate is dehydrated in a sense. So what we have obtained? Phosphoenol pyruvate. Now the 10th and the final step. This is the final step, 10th. Here the phosphoenol pyruvate is converted to pyruvate with the help of pyruvate kinase. This is the final step. Here the phosphate group is attached to the 2 carbon. Here phosphate group is attached to the 2 carbon of phosphoenol pyruvate and it transfers to a molecule of ADP. It transfers to a molecule of ADP and it will yield ATP in the final step. So here we have 2 molecules of phosphoenol pyruvate. Okay, like we have in the previous one. So here also 2 ATP we will get. We will get 2 ATP here also. So till now what we have obtained here in the first step, here in the first one. Okay, in the first one what we have obtained, first one here, 1 ATP is dissociated. Okay, in the first step. It is used. Energy is used here. Okay. Now in the second and the third step also 1 ATP is used. So till third step 2 ATPs are consumed. In the seventh step the 1,3 bisphosphoglycerate is converted to 3-phosphoglycerate. Here 2 ATP is gained with the help of ATP. 2 ATPs are gained. Basically 2 atoms of 1,3 bisphosphoglycerate are present. So 2 ATP we will get. Likewise in the 10th one 2 ATPs are formed here. So finally this is the cycle we all know. Okay, this is the cycle we all memorize. This one, glucose, we have explained it stepwise. So 1 2 3 4 5 6 7 8 9 10, there were 10 simple steps. So we have explained each one of them in a brief way. You can write it in your own language as well. You can take screenshots and write it. So first one is glucose. Here ATP is utilized. Glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 bisphosphate, glyceraldehyde 3 phosphate. Okay, then 1,3 bisphosphoglycerate, 3 phosphoglycerate, 2 phosphoglycerate, phosphoenol pyruvate and pyruvate. These are the steps. So here like we have said, uh, step 1 and 3 2 ATPs were used. Step 7 and 10 2 ATPs each were utilized. Okay, 2 in each step, 2 in step 7, 2 in step 10. So you will be clear about how much ATP are utilized, how much ATP is used. So net visible ATP produced is 2. So finally what we will obtain by the cycle, 2 ATPs we will get finally. So this is the basic idea of the glycolysis cycle. We also know if glycolysis won't work, we will not be able to maintain the sodium phosphate pump and eventually it will die. So the main purpose of glycolysis, it is to provide the pyruvate so that it can go for the TCA cycle. TCA, what is the full form? TCA, trichloroacetate acid. Trichloroacetate acid cycle. The next cycle we will go. So the pyruvate will go from here and it will perform the TCA cycle, or we can say the Krebs cycle. Okay. So the glycolytic production of pyruvate, it reduces the cytosol by increasing the ratio of NADH. This is the purpose of glycolysis. This is the basic purpose of glycolysis. The breakdown of the glucose molecule into two smaller molecules is the main motive of the glycolysis cycle. So this was all about the glycolysis. I hope you all were able to catch the main point of glycolysis cycle and how the ATP is utilized, how the ATP is produced, and the net visible ATP at the end what we get. So here you can see like if you want to denote here, like here -1 ATP. Okay, here you can say -1 ATP, here +2 ATP, and in the last one here +2 ATP. So these two are utilized, so this will cancel. Okay, this and this equivalent, so finally we will get 2 ATP. This is the net gain of the ATPs in the glycolysis cycle. Now next we will try to understand the TCA or the TCA cycle and more cycles in the pathway. Okay, we'll try to understand. uh, so what is required in the glycolysis cycle. If someone asks you, you have to understand the glycolysis cycle. So what all things are required? So ATP is required, ADP is required, NAD is required, glucose is required, and different cytoplasmic enzymes are required. What are cytoplasmic enzymes? These hexokinase, phosphoglucoisomerase, phosphofructokinase. Okay, these all are cytoplasmic enzymes we can say. So this was all about this video. I hope it was, no, it was easy to understand, and we'll try to come up with more such videos.